and I have created this project for the Gemma for Good Hackathon 2026, which is live here on the Kaggle website. So here's the pitch of my project. In the year 2024, the Nobel Prize in Chemistry was awarded to the team behind this project called AlphaFold, and they deserved every bit of this Nobel Prize. So for 50 years, scientists had one of the hardest unsolved problems in biology. So given a protein's genetic sequence, what shape does it fold into? Getting that shape wrong mean drug failed. Diseases went untreated. Billions of dollars were wasted. But this AlphaFold single-handedly solved this problem in one shot. It predicted the 3D structure of over 200 million proteins. Basically, every protein known to human science with accuracy that matches expensive laboratory equipments. So it was genuinely one of the greatest scientific achievement of lifetime. It is one of the greatest human exploration in the field of biology and the chemistry. But this left us with one problem. This problem that nobody has solved since then. AlphaFold tells you the shape of the protein, but it does not tell you what the protein does. And this is the actual question that matters for medicine. Does it causes cancer? Does it trigger Alzheimer's? Can you make a drug that blocks it? And right over 100 million of proteins have a predicted structure but zero known function. Scientists call this dark proteome. It is not small problem. Over 30 to 40% of human proteome is completely in the dark. A researcher trying to understand uh, just one of these proteins would take around 6 to 18 months of reading papers, running experiments, and often still not knowing the answer. So, this is what my project, which is called AlphaFold Gap Filler. I have named it Gap Filler to make sure that it just fills that gap between what has been lacking behind since the 2024 Nobel Prize winners. So what my project does, it takes any uncharacterized protein, something with no non-function, no literature, no nothing, and it tells you what it likely does. So it not exactly tells you what it does, but it uh, gives a hypothesis, a prediction on what the function of the protein will be with a confidence score, and which can be used like in the which existing drugs might target it, and when experimented to run first it confirmed. So for example, for this project we have used a protein. Let me tell you this protein. Here it is, Q8IYJ2 or C10 or F67. So it is a mitochondrial human protein. So for example, now if someone want to know the function of this protein, they have to run, let's say, hundreds of tests in control environment, because as of now we do not know anything about these proteins. But what if my project AlphaFold Gap Filler, if it can predict with a confidence score, if it can predict the function of this protein, then the numbers of experiment that we have to perform will become very limited. So for example, rather than performing 100 random experiment on this protein to get to know the function of this protein, we have to run maybe 2 to 3 limited environments where we can target the function of that protein, assuming the target the function of that protein, and we'll be able to test if it is actually the work it performs as it is predicted by my project AlphaFold. And all this can run in under 7 minutes. So in under 7 minutes you can bypass months of research and billions of data. So for our project, the target protein I have choose is Q8IJ I by J2 or C10 over ORF67. So it is a human protein sitting inside the mitochondria, the powerhouse of the cell. Nobody knows as of now what it does. PubMed returned 41 characters of literature about it, and this is less than even 1% to one sentence to know what the function of the protein is. It has one blast homologue, which is itself and it is, as of now, it is not associated with any diseases in science. So this is a complete unknown protein. This protein is a complete unknown protein as of now. Now, I have already run this pipeline, and let me tell you this pipeline. This is here everything start like this is the setup and extraction of the files from this project. This is installing the packages that are required for our project. It checks the import done, and here is the part where I load the Gemma for $2 billion 2 billion parameter model. As you can see, it is completely loaded and use this memory 10.2 GB. And here is our analyze the truly uncharacterized protein. So here we are analyzing the dark proteome target, which is this one. Now, as you can see, it has generated a result. Now, how this result is generated and what it does, let me explain you. So it is pulling the revolutionary relatives from BLAST, checking the genomic neighborhood of this protein, querying the interaction network, like what other protein does this protein interact to, and also pulling the structural data of that protein from the very first project I told, AlphaFold, which won the Nobel Prize in the year 2024. So it is also pulling the structural data from AlphaFold and also finally searching the literature of this protein, and it is doing all this work simultaneously. So, for example, in real life, if researchers have to do the same project, they have to get together a team of maybe 5 to 10 individuals who will work on this project separately, work on all the 5 parameters separately, and then they have to come together to merge this. But this might lead into the lack of attention to some details, which can affect our prediction of the function of that protein. So what my project does is it did all the things that I mentioned, all the parameters, and after completing, it gave a primary function hypothesis. Like it gave a hypothesis of what this protein do. So uncharacterized protein C10ORF67 acts as a mitochondrial adapter protein involved in regulating innate immunity signaling, specifically functioning as a scaffold that bridges components necessary for inflammatory responses at the mitochondria, which potentially modulating host defense against pathogen or stress signals leading to neuronal damage. So this sentence is actually years of research put into this protein and hundreds of thousands of dollars put into this protein research, and this project does with in just maybe, let's say, under 30 minutes. And also it gave us a confidence score, how much confidence it has on this hypothesis, which is 64.8%. Now what a researcher would need to do is he have to run a very limited experiments with control environment to check if this protein actually does this. If it does this, then this again my project becomes a vital, like a worthy candidate in the Nobel Prize. So maybe I can win a Nobel Prize for this project. I don't know. Maybe. So it has created a biological process confidence score, and now further, if we go further, it has displayed the full report. Here, the full report has been displayed, like how it came to this conclusion. Rather than just watching the conclusion, here AI generated the function hypothesis, and it is also giving us the reason why it came to this conclusion, also giving the evidences which can be put to work. Now I have also added a uh, validation cell. So what it does is it validates the function of an existing protein. Why I added this? This is you can consider it a checkup before believing the actual, before believing the actual result of the unknown protein. So this validation, it runs a known protein, a protein that we know everything about, and it generates the result of that protein, what it does after the research. So if this tells you the exact function of that protein, which we know as of now, then we can we can be confident that yes, the cell is working completely fine, and whatever it has generated, like whatever result it has generated, we can believe in that because it provided the function of a known protein as well. Now, here is the drug repurposing dependency. So it is running the drug test on the repurposing of this hypothesis. So what this does is, what it does is it tell you the biological process confidence which is high for this, and uh, let's move to the next part. This is not quite explanatory. You will just read it like how this drug repurposing actually helps. And let me tell you the next cool part of this project, which is bold Z2. So let me scroll down to the bold Z2. Yes, here it is. So what is bold Z2? It is an open source AI model from MIT, from Massachusetts Institute of Technology, which predicts the actual binding strength of each drug to this protein. So it is an AI model which is like produced by the MIT alumni. And let me tell you the function of this. So as you can see, this is the name of a existing drug, SB203580, and it uh, and this tells you the binding affinity of the protein that we have through all the research that we generated, and through all the research generated by the Boltz 2 model. It has created a resultant binding affinity of the both the proteins, which can be used in synthesis of drugs. So for example, this is a drug which might be able to solve the problem that we are targeting with our unknown protein. So this cell creates the percentage score of the binding affinity of both the proteins, and it can run the same result for thousands of unknown drugs, and it will rank them according to the binding affinity. So how well both the proteins will bind together to create a single drug. Now, why it is only possible with Gemma 4, I'll tell you, because of the long context window of the Gemma 4 model. It has 1 million token context window, which is great because it will give him the complete understanding of what we are trying to create. So eventually, Gemma Force 1 million token window makes this possible for the first time. And I have fed this cell with all the five evidence streams into single inference call. So Gemma holds all this together, reasons across all these parameters, and produces a hypothesis the way an expert scientist would do. No other model as of now can do this, and I'm very sure about this. So now, how it can be used, like how this project can be used further. So there are 100 million uncharacterized protein in AlphaFold database. At the current rate of manual research, it would take humanity thousands of years to understand them all. Every one of this protein is a potential drug target. Every dark uh, protein characterized is a potential treatment for a disease that currently has none treatment. So the neurodegradation drug market alone is $22 billion, drive almost entirely by targets that do not yet exist. So we are building the Atlas. We are building an encyclopedia for the protein, a continuously updated map of dark proteome where every newly deposited alpha fold structure automatically analyzed connected to existing drugs within hours, and the next version runs 10,000 proteins simultaneously after this. So this can be further scaled to hundreds of thousands of protein structured studied daily. So maybe in less than a year, we will be able to find the cure of some of the most dangerous diseases, fatal diseases, and maybe able to stop them. So AlphaFold told us what protein does look like. What I have built tells you what this protein do, how it can connect with diseases, how it can bind with the existing drugs, so that we can generate better drugs for the treatment of the diseases. So please have a look through of it. I will submit a written write up for the clearer understanding. And thank you so much. Bye bye.